Step 1. Determine whether genetic engineering is the most effective way to solve a plant's problem, such as a disease or damage from pests. For centuries, traditional plant breeding has been used to fight pests, environmental stress, such as drought, as well as to improve taste and nutrition. A powerful new tool, biotechnology, was now available to fight back. Farmers and researchers turned to genetic engineering beginning in 1985 for help. Step 2. Identify the gene or genetic material that could solve the problem, like providing resistance to the disease or pest, and study the genetic makeup of the plant that needs the trait. Researchers and farmers had to find a way to make the papaya plant resistant to the deadly virus. PRSV resistance was the genetic trait needed to solve the problem. Researchers knew that if plants contained certain genetic material from the virus within their own DNA, they were protected. If they could insert a copy of this gene from the virus itself into the papaya's genetic makeup, the plants would be protected. Step 3. Copy the trait from a donor organism and implant it into the plant's DNA. Researchers isolated a PRSV gene from the virus itself. Then they needed a way to get the genetic material into the papaya plant's own DNA. Researchers deployed a commonly used technique in biotechnology called transformation, a process by which a copy of genetic material from a specific trait is inserted into cells. There are many ways to transform a cell. In this case, the researchers used a high-speed particle bombardment method. Through a high-pressure insertion device, the gene from the virus was inserted into the DNA of the papaya seed. The genetic material made the plant resistant to the ring spot virus, just as a flu shot makes people resistant to the flu. The researchers were able to successfully transform 17 plants. These plants contained this gene from the virus and produced a protein from the virus that helped the plants become resistant to the PRSV infection. Step 4. Plant the new seed and test it.